Good morning, everyone. Today we are going to discuss sustained release dosage forms, an important topic under controlled and the modified drug delivery systems. So, before discussing the formulation approaches, we should understand why sustained release is required. So, when a conventional immediate release tablet is administered, the drug is rapidly released and absorbed. So, this produces a relatively rapid increase in the plasma drug concentration here. So, after reaching the peak concentration, the concentration gradually decreases because of distribution, metabolism, and excretion. So, therefore, repeated administration is required to maintain the drug concentration within the therapeutic window. So, the objective of the sustained release formulation is to extend the duration of the action, that is drug action, by releasing the drug slowly over a prolonged period here. So importantly, sustained release does not necessarily mean that the drug is released at a perfectly constant rate here. So rather, the formulation is designed to slow the drug release compared with an immediate release dosage forms here. A sustained release dosage form is a formulation that releases the drug at a slower rate than the conventional dosage forms and consequently prolong the therapeutic effect here. So the important concept here is rate control. So in an immediate dosage form, that is immediate release dosage forms, the formulation generally does not provide the significant control over the release rate here. So the drug dissolves rapidly, becomes available for the absorption and produces its pharmacological effects here. So in sustained release, what will happen? The formulation variables such as polymer type, polymer concentration, drug solubility, particle size, porosity, membrane thickness, and formulation geometry are manipulated to retard the drug release. Okay, so by editing all these factors, we can manipulate the drug release rate here. Okay, so the ultimate objective is to maintain the drug concentration within the therapeutic range for prolonged period of time, that is longer period of time, and thereby reduces the frequency of administration. Okay, again I'm repeating this line. So the main objective of this formulation is mainly to maintain the drug concentration in the therapeutic window for longer period of time. Okay, underline that point. So by creating this type of formulation, the ultimate goal is to reduce the frequency of administration. Okay, so why do we need the sustained release? So let us now understand the pharmacokinetic problems that sustained release systems attempt to solve. So after administration of an immediate release dosage form, so the plasma concentration usually increases rapidly and reaches a maximum concentration here, that is called as Cmax, at a particular time. So that time is noticed as Tmax. Okay, so after this peak, the concentration will again decreases. So if the concentration falls below the minimum effective concentration, so the drug may no longer produce as the desired therapeutic effect here. So if another dose is administered too early, so the concentration may rise excessively and approaches or exceeds the toxic concentrations here. So therefore the repeated immediate release dosing can produce the peak to trough fluctuations here. Okay, so sustained release formulations attempt to reduce these fluctuations by extending the drug input into the systemic circulation. Okay, so what is therapeutic window? So this slide illustrates the concept of the therapeutic window. So the minimum effective concentration, or MEC, represents the approximate concentration above which the desired therapeutic effects can be obtained. Okay, so the minimum toxic concentration, or MTC, so represents the concentration above which the toxic effects are more likely to occur. So ideally, plasma drug concentration should remain between these two limits here. A conventional dosage form can produce the large fluctuations between the peak and the trough concentrations here. So a sustained release system attempts to produce a smoother concentration time profiles here. So this is particularly valuable for drugs having a narrow therapeutic window. So provided the drug possesses a suitable pharmacokinetics and the biopharmaceutical characteristics for the sustained delivery. Okay, what are the objectives of the sustained release? So the main objectives can summarized into several points here. So first, we want to prolong the duration of drug action, so that is the main objective. So second one is we want to reduce the frequency of administration. Third, we aim to reduce the fluctuations in the plasma drug concentration. Fourth, sustained release can improve the patient compliance because the patient does not have to remember to take the medicines as frequently. Okay, so that's the another thing. And fifth, so maintaining the concentrations within the desired therapeutic range may produce the concentration related adverse effects. And however, we should remember that the sustained release is not automatically better than the immediate release. So the formulation must be appropriate for the drug, disease conditions, and the pharmacokinetic requirements. Okay. Sustained release versus controlled release. So this distinction is very important for the PG students. So the term sustained release and controlled release are sometimes used interchangeably in pharmaceutical literature, but technically they are not identical. Okay, so a sustained release system primarily aims to extend the drug release and duration of action here. A controlled release system implies a greater degree of control over the drug release rate. So identical approaches a predetermined release profile here. So in an ideal controlled release systems, the drug release may approach a desired zero order pattern here. So therefore, every controlled release systems can provide the prolonged drug delivery, but not every prolonged or sustained release systems provides the precise zero order release. Okay, so ideal drug candidates. So not every drug is suitable for the sustained release formulation. So the drug should preferably have a suitable dose size. Okay, so that's the minimum requirement. So the drugs requiring the extreme large doses may be difficult to incorporate into a particular sustained release dosage form here. So the drug should also have an appropriate biological half-life. So very short half-life may benefits from sustained release. So but the formulation must provide the sufficient prolonged release. Okay, so on the other hand, the drug with extremely long half-lives may not require the sustained release technology because the body already maintains the drug for prolonged period here. So the drug should also have a suitable absorption characteristics. If absorption occurs only in very limited region of the gastrointestinal tract, designing an oral sustained release systems becomes more challenging here. Okay, so important drug selection criteria. So when selecting a drug for sustained release, we consider several parameters. So these parameters includes like dose, half-life, absorption window, therapeutic index, solubility, stability, and pharmacokinetics. So the drugs with very high water solubility may diffuse rapidly from certain matrix systems. So very poor soluble drugs may show the incomplete or excessively slow release. So the drugs with extensive first pass metabolism may require the additional consideration because prolonging gastrointestinal residence does not necessarily guarantee the prolonged systemic exposure here. So we therefore have to evaluate the entire ADME profile, that is absorption, distribution, metabolism, and excretion before deciding on a sustained release approaches. So classification of the sustained release systems. So sustained release systems can broadly be classified according to the mechanism controlling drug release here. So the major approaches includes diffusion control systems, dissolution control systems, osmotic systems, ion exchange systems, and chemically control systems. So in practical dosage forms, these mechanisms may also operate simultaneously here. So for example, a polymeric matrix tablet may dissolve drug dissolution followed by diffusion through the hydrated polymer network. So therefore the actual release mechanism is often more complex than a single theoretical model. Okay, so matrix systems. So matrix systems are among the most widely used approaches for the sustained release tablets. So in a matrix system, the drug is dispersed through a continuous matrix forming material. So the matrix may be composed of hydrophilic polymer, hydrophobic polymer, wax, or other suitable materials. So when the dosage form comes into contact with the gastrointestinal fluids, so the drug is released through the mechanisms such as diffusion, erosion, dissolution, or combination of any of these. So the matrix tablets are attractive industrially because they are relatively simple to manufacture compared with some sophisticated reservoir systems. Is it clear? And next is the hydrophilic matrix systems. So now let us examine the hydrophilic matrix. So a common example is HPMC, that is hydroxypropyl methyl cellulose. When the tablet contacts gastrointestinal fluid, water penetrates into the polymer matrix. So the polymer hydrates and forms a gel layer around the tablet here. So this gel layer becomes the major barrier controlling drug movement. So initially the water penetrates into the tablet and the drug dissolves. So the dissolved drug then moves to the hydrated polymer network towards the external medium. So at the same time, the polymer chains can gradually relax and erodes from the system surface. Okay, so therefore the drug release from the hydrophilic matrices may involve both diffusion and the polymer erosion. And next is the hydrophobic matrix. So in a hydrophobic matrix, the water penetration is more restricted. So materials such as certain waxes or water insoluble polymers can form the matrix here. So the drug dissolves within the pores or channels created inside the matrix and subsequently dissolves through these pathways. So the release rate depends on the factors like polymer concentration, porosity, drug solubility, particle size, and the matrix. Increasing the amount of the hydrophobic material generally increases the diffusional resistance and can retard the drug release here. Okay, and next is the reservoir system. So another important approach is the reservoir system. So here the drug is present in a core and the core is surrounded by a rate controlling polymer membrane, and the membrane acts as a major barrier to drug transport here. So the drug release may occur by diffusion through this membrane. So the advantages is that the membrane characteristics can be carefully manipulated to control the release. Okay. However, reservoir systems require greater manufacturing control because defects or damage in the membrane may alter the release behavior here. And next is the diffusion controlled drug release. So diffusion is one of the most important mechanism in the sustained release formulations. So according to the Fick's law, the drug movement occurs because of a concentration gradient here. So in a simplified form, the equation is as here: J is equal to minus D into dC by dx. Here J represents the flux of drug and D represents the diffusion coefficient and dC by dx represents the concentration gradient. So the negative sign indicates that diffusion occurs from region of higher concentration towards the region of lower concentration. So in pharmaceutical systems, the polymer structures can significantly reduce the effective diffusion coefficient and therefore slow drug release. Dissolution control systems. So in dissolution control systems, the rate at which the drug or its controlling barrier dissolves determines the release rate here. So there are two broad possibilities. So the drug itself may be formulated in a poorly soluble form and thereby slowing the dissolution. Alternatively, the drug particles may be surrounded by a slowly dissolving coatings. So as the coating gradually dissolves and the fresh drug surface becomes exposed here. So this approach is particularly useful when the formulation can achieve predictable dissolution behavior under physiological conditions. Is it clear? And next is the osmotic pump systems. So osmotic systems are highly sophisticated drug delivery systems. So the basic principle is generation of the osmotic pressure inside the dosage form. Okay. So the system typically contains a drug compartment and an osmotic agent surrounded by a semi-permeable membrane. So when water comes from the gastrointestinal environment enters through the semi-permeable membrane here, so this produces the pressure inside the system. Okay, so the resulting pressure can push up the drug formulation out through a precisely engineered delivery orifice. So one important advantages of the osmotic system is the drug release can be made relatively independent of several physiological variabilities compared with the conventional matrix systems. Okay, and next is the ion exchange resin systems. So ion exchange systems use charged polymers called the ion exchange resins. So the drug is bounded to the resin through the ionic interactions. When the drug resin complex enters the gastrointestinal tract or gastrointestinal fluids come in touch with the gastrointestinal fluids, and the ions present in the surrounding media can exchange with the drug. Okay, so the drug is consequently released gradually here. So because of the exchanging of the ions, the drug will release gradually here. So the release mainly depends on factors like resin properties, degrees of cross-linkings, particle size, ion strength, gastrointestinal pHs, so all this. So these systems get particularly useful for controlling the release of the ionizable drugs. So these systems can be particularly useful for controlling the release of ionizable drugs. Microencapsulation. Microencapsulation is the another important technique. So here the individual drug particles or droplets are surrounded by coating material to form the microcapsules here. So such capsules are called the microcapsules. So here the drug core may be in the form of a solid or a liquid. Okay, so this solid or liquid is get turned into a solid form again. Okay, so by using of the coating material, we are converting the solid or a liquid droplet into a solid form again. That too in a micron size. So that's why these are called as the microcapsules here. So the coating acts as a diffusion or a dissolution barrier here. So by changing the polymer types, the coating thickness, permeability and the particle size, the drug release rate can be modified here. Okay, so microcapsules are subsequently be incorporated into the capsules, tablets or suspensions. This approach also allows different populations of particles with different release rates to be combined in a single dosage form. That is, we can edit the release rate by using different type of formulation behavior. So here we can achieve the different release rates by using a combination of different polymers here. So here how we can achieve this means we can change the formulation factors, so formulation concentrations, so in order to produce the release rate at a particular time. Okay, so suppose if you prepare some batch of particles to release at a particular time, some to be released at a next level. Okay, so by increasing the concentrations are by editing the formulation concentration factors, we can achieve this. Okay, so this approach also allows the different population of particles with different release rates to be combined in a single dosage form. So here how we will prepare this means, suppose if you take some example like in a Coldac tablet, so Coldac capsule, right? So in Coldac capsule, did you observe means it has a different sort of colored particles are there. So those particles are designed are formulated in such a way that they will release in different time intervals to achieve the sustained release here. So the main aim or main goal of our formulation is to prolong the release rate here. Okay, so that too in a therapeutic window. Okay, we already manufactured some formulation. So here how the formulation is designed means, suppose if you take a Koldak tablet, in that tablet it has different type of particles we already discussed now. So like if there are three type of formulations are there means we can combine, so some release at a particular time. Suppose if there is a yellow color particles are there, if they can release at a particular time, let us will take some estimation like they will release the drug in 15 seconds or less, otherwise 15 minutes. Okay, after 15 minutes it will be out. Okay, so after 15 minutes the level of the therapeutic curve will again fall down. Okay, so we doesn't allow to fall the curve here. Okay, so we have to, in order to maintain the curve rate, in order to maintain the, so the curve rate itself indicates that it is showing the action. Okay, therapeutic action. Okay, so the main aim of our formulation is to maintain the therapeutic level. Okay, so if you take the formulation yellow color particles, so they can release the drug, the ability of the formulation is to release the drug up to 15 minutes only. After 15 minutes its action will be completed. Okay, so after that next sort of pattern, that is our white color particles will start producing the action. Okay, after 15 minutes here the curve is not falling down here, observe. So after yellow color particles, white color particles coming to the action next. Okay, so they can produce action up to 20 minutes. Okay, after 20 minutes again the curve is ready to fall. So next pink color particles will act. So here the curve is maintaining in the straight line pattern in the therapeutic window like this. Okay, so here we are using three types of formulations. So first batch is used to release the drug in a particular time. So after, before it's getting declining its action, the next batch will come up. Okay. So like that we have to maintain the action in a prolonged way by using this sustained release formulations. Okay, so the drug release kinetics. So now we come to the very important M.Pharm level concept, that is drug release kinetics. So we need the mathematical models to understand how the amount of drug released changes with the time. Okay, so the major models includes zero order kinetics, first order kinetics, and the Higuchi diffusion kinetics. So in addition, the Korsmeyer Peppas model is widely used for analyzing the drug release from the polymeric systems here. So these models do not simply give us an equation here. So they help us to understand the probable mechanism controlling the drug release. Okay. Zero order release. So the zero order release means that the drug is released at a constant rate here. So the mathematical expression is Qt is equal to Q0 plus K0 into T. So where Qt is the amount of the drug released at a time, at a particular time t. So Q0 is the initial amount released. Okay, K0 is the zero order release constant and t is the time here. So if Q0 is 0, so the equation becomes Qt is equal to K0 into T. Okay, graphically a plot of cumulative amount of the drug released versus time gives a straight line here. So the zero order release is highly desirable because it can theoretically maintain a relatively constant input of drug into the body here. So however, the achieving the perfect zero order release in an oral dosage form is technically difficult. Okay, and next is the first order release. So in first order release, the release rate depends on the amount of the drug remaining in the dosage form. So as the amount of the drug remaining decreases, the release rate also decreases here. So the model can be expressed in the logarithmic form as log Q is equal to log Q0 minus K1 into T by 2.303. Okay, so the exact form depends on how Q is defined. So the important concept is that release is concentration or amount dependent here. So the first order behavior is frequently observed in systems where dissolution or diffusion is properties to the remaining amount of drugs. Okay. Now next is the Higuchi model. So the Higuchi model is particularly important for the matrix type systems where diffusion is a major mechanism. So the classical Higuchi relationship is Q is equal to K into H square root of T, where Q is the cumulative amount of the drug released per unit area and KH is the Higuchi diffusion or release constant here. The important feature is that the square root of time relationship. So if a plot of the cumulative drug release against the square root of the time gives a linear relationship, so the diffusion control release may be indicated here. So however, the assumption of the Higuchi model must be considered before interpreting the results. However, the assumption of the Higuchi model must be considered before interpreting the results data. Okay. Korsmeyer-Peppas model. So the Korsmeyer-Peppas model is particularly useful when the release mechanism is not clearly identifiable or when more than one mechanism may be involved. Okay, so the equation is mt by m infinity is equal to k into t power n. Okay, so here mt represents the amount of drug released at a particular time. M infinity represents the amount of the drug released at the infinite time. So K is the kinetic constant related to the dosage form and the drug release characteristics here, and N is the release exponent. So the value of N help us understand whether release is predominantly controlled by the diffusion, polymer relaxation, erosion or combination of mechanisms. So for accurate interpretation, the applicable geometry and the selected data range must be considered here. So factors affecting the drug release. So several formulations and the physiological factors influence the sustained release here. So from the formulation side, we consider polymer concentration, polymer molecular weight, viscosity, particle size, porosity, drug solubility, drug loading, compression forces and the coating thicknesses. So all these factors are needed to be considered. So from the physiological side, factors includes GI pH, fluid volume, motility, food effects and the gastrointestinal transit time. Okay, so therefore the sustained release formulation is not simply a matter of adding polymers. So the formulation scientist must understand how the drug, polymer and the physiological conditions interact here. So effect of the polymer concentration. So the polymer concentration is one of the most important formulation variable here. So if you increase the concentration of the hydrophilic polymer such as HPMC, the hydrated gel layer generally becomes a thicker and provides the greater resistance to drug diffusion. So consequently, the drug release rate may decreases here. However, the relationship is not always linear. Very high polymer concentration can alter the tablet swelling, erosion and the diffusion pathways. So therefore, the polymer concentration must be optimized experimentally rather than selected simply on the assumption that more polymer is always better like that. Okay. Effect of the drug solubility. So the drug solubility has major influence on release. So higher water soluble drugs can dissolve rapidly and may diffuse quickly through a hydrated matrix here. So poorly water soluble. So the poorly soluble drugs may show the slower dissolution and therefore slower the release here. So however, the extremely low solubility can lead to incomplete release. So therefore, the formulation scientist must balance the drug dissolution and the polymer control diffusion here. So sometimes different grades or combination of the polymers are also used to obtain the desired release profile. Evaluation of the sustained release dosage forms. So evaluation begins with the normal pharmaceutical quality tests. For tablets, we assess generally the appearance, thickness, hardness, friability, weight variation and the drug content. So but for sustained release products, so these tests are not satisfied here. So the most important test is the in vitro drug release study. Okay, so we need to determine how much drug is released at different time intervals and whether the observed profile matches the desired release specification here. So in vitro dissolution testing. So dissolution testing is central to the sustained release dosage form development. So the appropriate USP dissolution apparatus like medium, volume, temperature, agitation conditions are selected according to the formulation and product requirements, according to the IP specifications or USP, whatever it may be. So the samples are withdrawn at a predetermined time points. So the drug concentration is analyzed using an appropriate analytical methods like HPMC, UV visible spectrophotometries. So the cumulative percentage of the drug release is then plotted against the time. Okay, so the resulting release profile can be compared with the target profile and analyzed using mathematical models. Okay, in vitro in vivo relationship. So an important advanced concept is the relationship between the in vitro dissolution and in vivo drug absorption. So an in vitro release profile is useful, but ultimately the formulation must provide the desired therapeutic performance in the patient. Okay, so if the dissolution is the rate limited step for the absorption, an in vitro release profile may correlate reasonable well with the plasma concentrations here. So this can contributes to an in vitro in vivo correlations or IVIVC. So a successful IVIVC can reduce the need for the extensive clinical testing during certain formulation development changes here. So depends on the regulatory requirements. So what are the advantages? So let us summarize the major advantages here. So the sustained release systems can reduce the dosing frequency. That's the first utmost advantage, and thereby improves the patient compliance here. So in order to maintain the drug concentration for a longer period of time, so reduce the peak to trough fluctuations can be designed. Okay, so potentially reduces the concentration related adverse effects also, and improves the convenience and provides the prolonged therapeutic actions here. So these benefits are particularly valuable in chronic diseases where a long-term medication is required. Okay, limitations. So however, the sustained release systems also have limitations. So they are generally more complex to formulate and manufacture, so they can be more expensive also. So the dose adjustment is less flexible than with immediate release dosage forms. So if the formulation fails and releases a large amount of drug rapidly, so the drug dose dumping may be occur. So food and gastrointestinal conditions may sometimes alter the drug release here. So also the sustained release formulations may not be suitable for drugs with unsuitable pharmacokinetics or the absorption characteristics. Okay, dose dumping. So the dose dumping is particularly important safety concern here. So the dose dumping refers to the unintended rapid release of the drug from a formulation designed for a prolonged release. Okay, so this can result into an unexpectedly high systemic drug concentration. So the consequences can be serious, particularly for drugs with a narrow therapeutic index. Okay, so therefore the sustained release products undergo rigorous formulation development and dissolution testing to ensure that the release mechanism maintains the robust and expected physiological and the mechanical conditions. Okay, and next is the industrial manufacturing. So from an industrial perspective, sustained release tablets may manufactured using in a conventional processes such as direct compression only, wet granulation by dry granulation. So depending on the formulation, we will choose the method here. So the critical variables includes the polymer distribution, granule properties, compression forces, tablet porosity, and the coating characteristics here. So for coating systems, the coating process must provide uniform film thickness. Okay, so it's a highly skilled process. So processes parameters such as spray rate, inlet temperature, atomization and the drying conditions can influence the coating quality here. Okay, so maintaining the uniform film thickness of the particular tablet is highly important. Okay, so therefore the sustained release manufacturing requires the strong control of both the formulation and the process variables here. Okay, formulation optimization. So in modern formulation development, the optimization is often performed systematically here. So important independent variables may include polymer concentration, polymer viscosity, drug to polymer ratio, compression forces, and the coating weight gain. Okay, so the dependent responses may include percentage drug release at a specific time points, dissolution rate constant, similarly factor, and physical tablet properties. So designing of the experiments or DOE can be used to identify the interactions between the formulation variabilities and the development in a optimized formulation with the predefined release profiles here. Okay, so to conclude, the sustained release dosage forms are designed to prolong the drug release and the therapeutic action here. So the formulation, the fundamental objective is to control the rate at which the drug becomes available for the absorption. So the major approaches includes the matrix system, reservoir system, dissolution control system, osmotic systems, ion exchange systems, and the microencapsulation. So the drug release mechanism can be studied using a mathematical model such as zero order, first order, Higuchi, Korsmeyer, Peppas models. So for an M.Pharm formulation scientist, so the key point is that the successful sustained release requires the integration of pharmaceutics, polymer science, drug release kinetics, pharmacokinetics, dissolution testing, and the formulation optimization. It is not depending on single method. So all these are needed to be included in your formulation. Okay, so while you are designing, so all these parameters are needed to be integrated together. Okay, so therefore the sustained release formulation is not merely about solving the drug release here. So it is about designing in a scientifically controlled dosage form that provides the predictable therapeutic performances. Okay, so thank you for watching.